So, good afternoon, ladies and gentlemen. Thank you for coming to attend the 30th Instruct seminar series, Structure Meets Function. Today, it will be totally dedicated to artificial intelligence, which is a very important area for Instruct, and that is already being materialized in one of our most important projects called Fragment-Screen. So today we have three speakers. One that will present us about a specific project in the framework of artificial intelligence, and then two more dedicated to artificial intelligence applications, both in chemistry, structural biology, and more related to deep learning. So, without further ado, I will leave you with Beatriz Serrano-Solano, that I have the pleasure to introduce. So she will talk about AI for Life and the opening of the second open call. Beatriz, the stage is yours. Thank you for coming.
Thank you. Thank you very much, Carlos. And so thank you for the invitation to this seminar series. Yeah, so I'm going to introduce a bit what AI for Life is, and then also the different opportunities that we have for scientists. So AI for Life, it's a project that started in September 2022. So we are now halfway through it because it's going to last for 3 years. And it's coordinated by Uruguay Imaging, and the goal here is to bridge the gap between two different communities. There are the life science on one side, so all these researchers that produce a lot of data with many challenges to analyze this data. And on the other side, the computer science community that develops plenty of methods, but usually don't have access to the researchers to test those methods and apply that to improve research. So basically, with the project, we try to focus on these two communities, but especially in bringing them together through different opportunities. So from training, different hackathons for developers, challenges to compute for different solutions, but also open calls for life scientists to support them in their research. So the first open call we launched last year. We are going to be launching 3. So the project is going to last for 3 years, and we will launch one open call each one of them. And the first one was last year in February, and that was an open call for bioimage analysis support. We had some examples here of what people can apply for, and I will show you a bit more in a second. So basically, we received in total 72 applications. We were very happy about that. And you can see the different fields that people were applying from. So cell was very much expected, but we saw also plenty on agriculture, plant ecology, and so on. Also neuroscience, marine biology. This is just a field in which they self-identify themselves with, but at the same time, we also ask many other questions about what were those needs. So we have kind of a small landscape analysis of the different needs in life sciences that you can see in the link at the bottom of the slide. That's all the information is published on our website. So after the first open call, we selected 8 different projects. You can see here exemplary images from each one of them. They are different modalities of images, different type of problems, and different scientific disciplines as well. I will not go into details of any of them. Just wanted to show some examples for you to have an idea of the kind of projects that we are selecting. And like I said, we received 72. We could only select 8 due to the capacity of the project, but there were many other interesting use cases there. Sorry. So now we've launched the 22nd of January, we launched the second open call. This is going to be running this year, 2024. The deadline for application is on the 1st of March. We are here way more synthetic than the last one, but the type of problems are the same that we are welcoming. So basically, whenever a researcher is finding a challenge in an image analysis problem, and they think that deep learning can help with the research, they can just apply to this opportunity. So basically, we split into different areas here, and based on the experience from last year, the first example that we show here is the improving your image analysis workflow, and that's usually the most common one that people apply for. Sometimes people are even happy with the outcomes that they have in their pipeline, but they want to improve some parts. For example, if there's something that is manual, they want to have a fully automated workflow for the analysis and not just removing that manual part. We also provide here advice on training data creation. Some people presented some examples of data that was not properly labeled, and that would be an example for us as well. And the third one was a consultancy on available tools and solutions. So many people applied last time, also wanting to know more about the workflow or how deep learning can help them. So it's not just solve my problem, but also I want to understand how to solve my problem. And the last option that we have here is catering the problem to other experts. And that's because when all these different use cases that are not getting selected because we don't have the capacity, but they still want to find a computational expert that supports them, we have a section on the website that we're going to populate soon with different problems that could find an expert that is interested in supporting them. So it's going to be called the bartering corner, in which people just go there and try to find this biological problems that they can help with. So the timeline for this particular open call, it's here. So we started with the application phase that we are right now. As I said before, it's the 1st of March is the deadline. Then we will have a review phase. So first we have a kind of a check to see that the application is complete and everything looks good to be sent to the reviewers. And then we assign a couple of experts to each one of the projects to evaluate a bit more detail the proposal. And now this time we have something new that we didn't have last time. That's something that we've learned. So we introduce a consultation phase. And this is because many of the problems could be solved just having a call or a couple of calls with the applicants and providing them pointers to different tools and software solutions that already exist and could very, very well address their problem. So that's why we have this the consultation phase. Then we will announce the ones that are really in need for experts to work for a long time on that, and that will be end of May approximately. And then from June to November, there will be the whole time in which the different experts in AI for Life are supporting the applicants with their problems. The application, it's really simple. So we have here the whole process more or less debriefed. It doesn't take very long, really. So first, we want some basic application details, so that your email, your name, and so on, just to identify yourself and get in touch with you. And then the other one would be the project details. So give a title, the domain that I showed before in the word cloud, something very high level like that, the aim of the project, and then we will provide us a template, a slide template that people can fill out with some example image and very clear guidelines on how to describe the problem in a very simple way, and also identifying very well what the limitation is. Then we also want to know about the data that is available, because the idea here is to share it after the project together with the AI model that is trained to solve the problem. So we want to have a small sample so that the expert can look at it and look how feasible the image analysis can be there, the description, what it's a biological object that is being shown in the image, and the type of images, and then more information about the particular file format, the amount of data, if there is labeled data, how much of it, and so on. And then the other aspect that we want to know about is the analysis, if that was already analyzed or if it's just raw data that was recently produced. Also, which tool is used, and we also want to know if this is a tool that is typically used in the field. The reason for that is we want to have projects that have a high impact in the community and not just serving one individual. And then we ask also if the analysis is manual or fully automated. And the last part is about the impact and dissemination. Basically, we want to know if the applicants would be happy with sharing this information, to promoting their projects and talking about that, to make the solution known for other people in their communities as well. Yeah, so with that, I would like to give you here the links to the website. I will share the link to the slides later in the chat. But also, if you already know a lot about these topics and you want to review, there is also a form to sign up for this. And we have also talked about this in social media several times already. So if you know anyone that could be interested, please point them to these different links. If you have any questions, I will be happy the whole session, happy to answer you. But also, if you don't want to ask today and you have another moment to ask, you have an email at the bottom of the slide that you can also reach out to us with questions. And finally, one thing is that we have an AI for Life newsletter that is released quarterly. So please subscribe if you want to know about these opportunities and other training events and different things that we're organizing in AI for Life. And I think that's all from my side. Thank you very much.
Thank you very much, Beatriz. I think you presented a fantastic project that opens a lot of opportunities for something that is important for all of us, even if we are more molecular and structurally pleased. Of course, that image and imaging process is very important and one of the strongest areas for artificial intelligence and machine learning tools. So I don't see questions and answers at the moment, but as Beatriz said, feel free to contact her, prepare good proposals, and take advantage of this opportunity. And with this, I thank you, Beatriz, for your very good presentation. And I will call on stage the next speaker. That will be Thiago Rodrigues. Thiago is from Portugal, from the Faculty of Pharmacy at the University of Lisbon. Is actually about 200 meters from my office because I'm on the other side of the street, both from Instruct PT. So now he will tell us about machine learning to accelerate the chemical sciences with a structural biology twist, I'm sure. So Thiago, the stage is all yours.
Thank you so much, Carlos, for the kind introduction. And indeed, it's we are very, very close. So first of all, thanks for having me. I think the talk today will be at times slightly off topic at first, but there is always a connection to Fragment-Screen, and then you'll see why. But before actually going to the talk, I thought that maybe it's a good idea to give you an overview of our research interests. So we are currently a rather small but highly motivated and growing group. We are not pure data scientists. We are not pure synthetic chemists or medicinal chemists. We can do a little bit of everything. So that to say that we don't build machine learning models just because we can write the code and then try to find the applications for the code. There is always a chemical or biological motivation. So a proper research question, and then we try to answer the question. If it involves machine learning, that's fine. If it's only molecular docking or pharmacophore model, it's also fine. If it's purely chemistry, that's also fine. So we are not biased by the methods that we are employing. If we use machine learning methods, obviously data sets are very important. In our case, we are dealing mostly with small data sets. So we usually build our own data to build the models, and we usually run our projects in a sort of active learning cycle. So build the model, suggest the experiments, we run the experiments, feed the information back to the model, refine the model, so on and so forth. And so we have been applying this concept to synthetic chemistry, in particular the optimization of chemical reactions. This is something I will talk about today. More recently, and also in collaboration with IBM in Zurich, we have developed code to identify new chemical reactions. And this is also something I will briefly talk about today. More recently, we've become interested in applying machine learning tools also to accelerate nanotechnology, in particular nanoparticles with some sort of anti-cancer application. Essentially applying the same methods that we typically apply to small molecules, but now to nanoparticles. Our first applications in this field was essentially anomaly detection methods to study investment patterns. And this was published in the ACSNO paper. And I think about 1 week ago, we got another paper accepted, this one with a proper data set and building models to correlate nanoparticle compositions to in vivo endpoints. But I would say that more typically we have been interested in applications to chemical biology or medicinal chemistry, in particular the identification of drug targets or designing of small molecules, predicting false positive readouts, And today I will also show one application in this front. So for those of you who are not very familiar with machine learning, I would say that there are three key pillars in machine learning: the data sets, descriptors or representations, and algorithms. My own intuition about this is that you can have the best algorithm possible, but if you don't have the right data set, it's useless. And also if you're accustomed to doing research in the field, you will very well know that you'll spend 80 or 90% of the time getting the data, putting the data in a good shape, then to apply the algorithms. So there is a huge focus on the data sets, and this is really a problem in our applications. For example, in the optimization of reaction conditions, if we are interested in one specific transformation, it's not very likely that we'll find the exact transformation reported several times in the literature. So there are different ways of dealing with this. The way we decided to do was we just build our own data set. So we defined the search space, we performed 5 to 10 random reactions, collected the endpoints, and this can be conversion or yield, build the algorithm, predict the reaction yields for the remaining reactions in the search space, select a few for running, feed the information back to algorithm, refine the algorithm, so on and so forth, until we meet a certain end criteria. So this is the basic workflow. In this particular case of labmate.ml, we are using random forest, which from also some prior experience they tend to work very well with small data sets. So as our first use case, we decided to try to optimize this reaction, not because we were very interested in the product, although it's biophoric, and back in, I think it was 2014 or 15, I had worked with these kind of compounds as GPCR ligands. So there would be a biological link, but it was not exactly because of that we decided to use this tool reaction. It was mostly because we knew it was possible to get high yields, but we can find conditions where something is slightly off and then the yield goes down very sharply, very, very fast. So for us, it seemed the ideal use case for a preliminary test. It was not super difficult or straining for the algorithm, but it was also not very basic, okay? So these were the variables that we were allowed to optimize, all real values here, but in other use cases, there were both real values and categorical variables. So for example, solvent, the catalyst, the ligand. It's able to deal with all kinds of variables. And so we collected the 10 random reactions, all of them with poor yields. And then we decided to implement two selection strategies. One, fully exploitative. So it's the blue trace. So the goal here is to maximize yield as fast as possible. It doesn't really care about the uncertainty of the prediction. And then one more balanced approach, and this is actually divided in two phases. So for the first 10 iterations, you see a zigzag going up and down, right? So this is fully explorative. So it's not selecting the reaction based on the predicted yield. It's selecting the most uncertain. So this is, in principle, the most informative experiment for the model. And after iteration 11 up to the 20th, it was a balanced approach. So taking into account both the predicted yield and the uncertainty. And we were quite surprised to see that in the case of the blue trace, we were able to reach quantitative yield very, very fast. And this was sort of unexpected. And you can see that from at the first iteration, we obtained something that is 70+%. And this was quite surprising because we only had poorly performing reactions as our starting point. So this really shows that, somehow these reactions, these 10 random reactions, still have the blueprints for something that will work in the future. And this was quite interesting. And we decided to stick with this selection policy in the area examples. So here also to show how the hyperparameter set was changing with the different selection strategies. With exploitation, we don't really focus on improving the model, so the hyperparameters also don't change that much. And in the case of the balanced approach, which is much more explorative, then the hyperparameters of the random forest also change more. It's more dynamic. We did a bunch of comparisons also with human optimization. This is just one of them. Comparing the optimization of the Yugi reaction that you saw in the previous slide and to PhD level researchers. I'm not going to say that the machine learning method works well. This is a very limited comparison with only 2 people, but at least we can see that it seems to be competitive. So being the goal of freeing up brain power for more fulfilling tasks, I would say this seems to be a good alternative. And here, the chemical space projected with the reactions that were performed highlighted. One of the questions next is, can the conditions that we obtain through that optimization, can they be transferred to other set of building blocks? So what we did to answer this, and it's a not very comprehensive answer, so this was just one test, was picking up another set of building blocks that were reported in the literature, giving essentially no product. And so this was a paper from AstraZeneca that reported this chemistry. So with the optimized reactions or the reaction conditions transposed to this set of building blocks, we were obtained to obtain a little bit of products. Also, what we observed was that using the same workflow and the same search space, we were able to tune the method to optimize both the C2N or C4N coupling, in this case for this reaction. Other examples where we optimize chemistry, very common chemistry in small molecule, in medicinal chemistry, so reductive amination, olfination, but we also have glycochemistry and protein modification reactions here. And this is another paper where we synthesize a fragment-like natural product and compared LabMate, so the machine learning workflow, with kinetic modeling, which is the typical workflow in the industry for optimizing reaction conditions in process chemistry. And it was fairly competitive. Currently, we have a second version of the software. The big differences are the ways, or it gives more possibilities of encoding categorical variables. Before it was purely one-hot encoding. Now there are different ways. There is the possibility of changing the variables on the fly. So if in the middle of the optimization campaign, we decide to change the solvents or concentrations of the building blocks, whatever, this is possible. Before it was not. Now it's also possible to suggest reactions in batches, and before it's just one reaction at a time. And obviously this is not exactly how organic chemists work. So they usually put several reactions in parallel and then analyze them before deciding which ones to do next. So now it also suggests reactions in batches. And I think this is essentially the main differences. So this version 2 was co-developed with In Silico Medicine, and we are currently running pilot projects with AstraZeneca and Frank Lorius in Münster. Something that we are also very interested, and this is more recent, is identifying new chemical reactions. And the interest comes mostly from the typical use of machine learning is building models and then selecting the experiments with the highest label probability or the lowest variance in the predicted value. So it's mostly exploitation. And in this way, we are bound to find maybe new things or just link known knowledge in different ways. But it's questionable the novelty behind those findings. So we postulated in this chemical science paper that machine learning can be used more prominently as well for curiosity-driven research, and the goal here is to find the known unknowns and the unknown unknowns of science. So basically, in this paper, we also expand on an algorithm by Lee Cronin, essentially including concepts of serendipity, being targeted or untargeted, and how novel are those findings. This research was actually inspired by the observation here in LabMate. So this was purely curiosity-driven optimization. So just purely on uncertainty. And even though we were selecting reactions based on high uncertainty, we were able to obtain reactions that were performing already better than what we had initially. So maybe this was also, it was essentially the motivation for this. And when we are talking about chemical reactions, we essentially have a pool of building blocks, right? With different functional groups, and we can react them in many different ways. In some cases, you're going to find something that is very well established, but what we want to do here is to maximize surprise and find the unexpected reactions. So we have this project also with Theo at IBM in Zurich to identify new electrochemistry. This is our search space already with all the training data and the iterations done. Some of the reactions did not afford any compounds, some others did. We have already prioritized 6 reactions for profiling. Obviously, we will have to run lots of controls here, one of them being running the same reaction without electrochemistry, just to make sure that electrochemistry really is important for the outcome. Try to predict reaction products to see whether the compound that we isolate can be predicted, for example, from a machine learning tool, or again, give chemists the building blocks that reacted with one another. Please give us the reaction products and see whether it matches. So these are essentially the 3 controls that we are interested in. Also, what we think we have to reformulate in this case is how we measure how well our workflows are performing, because accuracy simply is not going to make the cut here. If we are selecting based on uncertainty, on average, our accuracy will be about 50%, right? So we decided to focus more on information theory principles. How much information are we adding to the system with every iteration? And so you can see here, the model, you're not able to see from the slides, but the model seems to be behaving almost randomly without being random. And this you can see from the Shannon entropy that we are adding to the model. So this is reactions, the amount of information of the 5 reactions that we selected at each iteration, it's always very close to 5. And then we randomly sample at each iteration as well a bunch of reactions and see how much information they would add to the model. And it seems to be lower. So this is it for synthetic chemistry applications. As I mentioned before, our typical use cases are more in medicinal chemistry. And one of the benchmarks that we run from time to time is comparing models for bioactivity prediction. This is something that we have done maybe half a year ago when we are expanding on this also with another collaborator. So the more traditional learning methods with the deep learning methods, here you can see 2 different flavors of multi-layer perceptrons, LSTMs. We also have graph neural networks and transformers, not here in the plot, but we have done that. And our observation is that it's still, for the low amount of data that we have, it's still the more traditional methods that seem to be the way to go. Okay. In the case of one application, we decided to, back in 2014, we decided to focus on hyperlongomine. So this fragment-like natural product that has Michael acceptors, so it's not very friendly from a medicinal chemistry point of view. It's also not very soluble in water. And our inspiration was this paper, which was later retracted, not because of data fabrication, but because of ethical issues. But what's very interesting about this natural product is it's very small, it has the Michael acceptors, and still it seems to be very selective for cancer cell lines, as we can see here from the transformed and the normal cell lines. So the normal cell lines, basically no activity. So machine learning to predict drug targets. So we know there would be some phenotypic effects. We had no idea about the modes of action. So using a self-organizing map, so another flavor of neural network, we predicted TRPC channels. So these are some one type of calcium channels. And we decided to test piperlongomine against a panel of TRPC channels. And again, we were very surprised to see that it was in this cell-based assay, it was pretty selective for TRPV2. This assay was outsourced from the beginning, so it would make no sense for us to implement all these assays in the lab. What we then did was implement TRPV2 to confirm the values that we obtained from the company that was hired. Different methods, independent of the method and the replicate, the IC50 was always between 1 and 5 molar. Because it's a microreceptor, we did the washout assays. It's either non-covalent or the off rate is very, very fast. And it's also non-competitive binder. So here we are stimulating TRPV2 with cannabidiol. And piperlongomine is acting as an antagonist. We collaborated with Vera and Pfizer on this. It took us, I think, about 2 years to get the cryo-EM structure of TRPV2 in nanodiscs, and indeed it's binding at a different place in comparison to cannabidiol. The resolution overall is not very good for structure-based drug design. Also at the binding pocket, I think it's 3.5 angstrom, the overall resolution between 4 and 5. So it's not very good for structure-based drug design. And you can see here the anticipated interactions would be these. But because, again, the resolution is not very high, we had to get additional proof of this. So what we did, mutated those residues, performed, again, the calcium influx assays, and indeed those residues seem to be important for binding. We also built a focused library of piperlongomine analogs, basically knocking out the Michael acceptors. The compounds are still active, but not as much as piperlongomine. So the working hypothesis here is the Michael acceptors are not crucial for activity, but they seem to lock the molecule in a certain conformation that promotes binding at least more efficiently. So at this point, we had already established the link between small molecule and TRPV2. The next point was in which cancer is this relevant? And with bioinformatics, we reached to the point where glioblastoma was the cancer to study further. We did a bunch of biochemical and cell-based assays before this, but I think in the end it boils down to the in vivo assay. Key challenges we had to solve: the poor solubility of piperlongomine, because glioblastoma is a brain tumor. Injecting subcutaneously the cancer cell lines simply is not a good model for this type of cancer. And again, which dose should we give to the mice? So very long story short, again, we developed a formulation, in this case a hydrogel, to implant at the tumor site just one time, and then it would slowly release piperologmin over time. So you can see here the cancer here, and then the hydrogel implanted on top of the cancer cell line. Two different cell lines, controls and treated groups, and you can see that the treated groups look much, much better in terms of tumor burden in comparison to the controls, and the Kaplan-Meier plots also with the tumor burden over here. This actually led to our startup company in 2019. Obviously, it's the stage that we are at. It is very different, so we had to reformulate a bunch of things. This is what I can show you. So the drug delivery system is completely different at the moment. Also does not affect healthy cell lines. It responds, so methylated and methylated types of cells that are resistant to the standard of care also respond to this treatment, and it's equipotent against glioblastoma stem-like cells, which is very interesting. Because everything had to be reformulated, so we had also to do the in vivo assays. This is with a suboptimal dose. Actually, I think it's 30% or 40% of the optimal dose, and we can already see a difference in comparison to the control. The histology here also showing the treated and the untreated cuts, and it seems to be really, really good. So as key messages for this talk, some of them I consider it's just my opinion. Obviously, a best method for us, and I think this is common sense, does not exist. It depends very much on the problem you're trying to solve, the data sets you have. In our hands, it has always been the random forest, the k-nearest neighbors, the very simple models, but it doesn't mean that it has to be like this every time. Our hope, and this is one of the reasons why we keep benchmarking from time to time, is that we are generating lots of data. So at some point, we will start seeing deep learning methods perform at least as well. The question is, the time it takes for training a deep learning model, does it really give the benefits in comparison to a simpler model that we can train in a very common laptop? So yeah, it's also not just the performance, it is also about the added value. Mostly because these methods don't take into account, or it's very difficult to take into account, the experimental uncertainty. So an R-square difference of maybe, I don't know, 0.2, how different it is if we are not taking into account the experimental uncertainty. So it's basically nothing. And the way we see these tools at the moment are just as assistants of chemical intuition. So avoiding the clearly bad experiments, those that we should definitely can avoid. And so in the end, accelerate the preclinical stage development of drugs. These works were developed obviously by many people from different teams. My team at Gonçalo's lab, both at IMM and University of Cambridge. Wong Kon for the nanotechnology papers. Daniel at Duke, he's been a long-term collaborator. We've been working together since our days at ETH Zurich. Same thing with Jimmy at In Silico Medicine, Taiwan. So the three of us were at Gisbert's lab at a certain point. Obviously, collaborators at IBM, AstraZeneca, also Faculty of Pharmacy, and Frank in Mercer. Funding is also very important, as you can imagine, especially when we go to in vivo assays. It becomes very expensive very fast, and so many different sources of funding must be acknowledged for that. And obviously you for your kind attention, and very happy to take any questions.
Thank you so much, Thiago, for this fantastic presentation.
Thank you so much.
We have time for 1 or 2 questions if someone in the audience wants to put it forward in the question and answers panel. Meanwhile, I would just like to comment that you actually explore well beyond structural biology. So you touched several fields, and it shows some very important things about artificial intelligence. First, that is not one size fits all solution.
Exactly.
That expectations have to be well calibrated.
Yeah, that is actually one of the challenges. So I come from a medicinal chemistry background. I was not trained as a chemoinformatics person or data scientist. I was trained in the wet lab, and I only started writing code during my postdoc a bit over 10 years ago at ETH. And so one of the key challenges, at least maybe it was even higher, maybe 5 years ago, was adjusting the expectations of people with the same kind of background I had for these kind of tools. So I saw 2 types of people that were very enthusiastic and people that certainly didn't believe in anything about machine learning. And so it's not all or nothing. And adjusting these, the expectations is certainly critical. Also to promote the tools. Yeah.
Absolutely. As well as benchmarking. You don't have an example like, I'm going to use my algorithm against the chess champion, for example. It's much more fluid.
Absolutely.
And also interesting to see that when you go to finding and to solving a structure, it can take a long time. Sometimes model, and I think this is something that could be important, is that you already have possibilities that can be tested very quickly.
Yeah, exactly.
Without going full on the structure.
Yeah.
Okay, so I have a question, and this is quite pertinent because machine learning is not something that comes off of the shelf. As you said, it takes time. It took years to code and to learn how to go this way. This comes from Edwin Lazo, and the question is: are these tools available? For example, GitHub.
Yeah. So yes and no. So for the benchmarking of bioactivity prediction, they will be available. Not at the moment because we are still running the project, but when we publish, everything will be available. So this is one. We have tools available for false positive predictions. I can share the GitHub on the chat if anybody is interested.
Yes, please.
For the self-organizing map, no, this is proprietary, so the tool is not available. For the reaction optimization, the first version is available. The second one, not so sure at the moment. We are still discussing because I mean, in silico medicine is involved. So we will have to see. The interest in both ends is to make it available. We just have to understand how and when. And for the reaction discovery method, it's definitely going to be available. Also not at the moment because we are still running the project, but this will be available. Yeah. And for the nanotechnology, the paper was accepted last week. And so the models and the data sets, this is actually one of the selling points of the paper. Actually didn't present here, but everything will be available. So the goal is to make it as freely available as possible. Sometimes it's not as easy because fortunately we have been collaborating with many companies, and then obviously there is IP management behind that. So it's not always very easy to navigate that.
Absolutely. Thank you, Thiago. We have time for one last question. This comes from Ether James, and is quite relevant and also touches something that is very dear to Instruct, and that is training. And this is, what are your recommendations for wet lab scientists that want to get into machine learning, that want to write?
I think in my case, it was It was just by chance. So as I mentioned, I started writing code during my postdoc. I went to that lab not really to learn code, but to be exposed to different research environment. I was doing chemistry for my projects and everyone else's projects, and at the same time that I was teaching my lab mates doing chemistry, I was also learning how to write code. But currently, there are a lot of very nice resources online. So Google Colab, notebooks, teaching the basics of Python, because everything that we do here is written in Python. Yeah, so I would strongly advise looking for those resources. Yeah, and go through them through the tutorials. It's also not just reading the tutorials, it's really writing the code and learning. There will be many failures and learning from those failures. So at least that's how it worked for me. So yeah.
Okay, thank you so much, Thiago.
Welcome.
Right, so for the last speaker of the session, I would like to call Enara Vigil on stage. So Enara comes from IBM, and they will be talking about accelerating drug target inhibitor discovery. And this time, it's not random forest, it's a deep generative foundation model. So Vigil, please, the stage is yours.
Yeah, can you see my screen?
Yes. Perfect.
Okay. Okay, so I'll be talking mainly about this paper, which appeared in Science Advances, accelerating drug target inhibitor discovery with a deep generative foundation model. But before I delve into the details of this paper, I would like to give some context into what foundation models are and how we came into solving this problem. And there was a lot of things that were done even much before we looked into drug target inhibitor discovery. So I would like to trace our path over the last few years. Okay, so some context into foundation models. So now everybody is familiar with ChatGPT. So ChatGPT is essentially a large language model, which is trained to predict the next word in a sequence. And because it is able to predict the next word, if it has to predict the next word, it has to create a model of the world around it, right? So even though the task it is trained on is to predict the next word, in order to do that successfully, it is able to create a representation that captures many aspects of the real world. But large language models and in general, like foundation models, are not just useful in language. In fact, this is the CEO, Greg Brockman from ChatGPT, OpenAI, saying that other modalities like image and biology could also be very useful. So what are foundation models? Foundation models are typically large models that are pre-trained on unlabeled data sets, and this could be many different modalities: language, time series, tabular data, images, and so on. And they leverage something called self-supervised learning. So what is self-supervised learning? In self-supervised learning, you assume that you don't have labels on the data, but you have large amounts of data, right? And so what you are trying to do is trying to predict some parts of the data using some other part of the data. So for example, in this example, you're trying to predict the future, right? So you're trying to predict the future sentence or the future words that appear in a sentence from what you have seen in the past. So that way, your model is able to build a good representation of the language. The same thing appears in images. There have been many different work where we try to mask some part of the image and then try to predict the masked part of the image. So the idea is the same. You don't need to have a large amount of labels. So you just look at your existing unlabeled data and try to build a very generalizable representation of the data so that it can be used for many downstream tasks, like, for example, text generation, machine translation, or classification. So the same breakthroughs that are happening in language, natural language, is also impacting molecule and machine material discovery. So for example, you have in large language models, you have letters, you have words, sentences, and finally the language. Similarly, for in chemistry, you have atoms, molecules, properties that you can use to predict based on your large models. So one of the first tasks that we looked into was, can we use AI to design new antimicrobials? So this was many years back. We started this project, I think around 5 years back now. So we were looking into antimicrobial peptides. So peptides are like these small, naturally occurring proteins. They're a crucial component of our immune system. So the question that we asked in the context of rising antimicrobial resistance was, can we build new peptides that would be effective against this drug-resistant bacteria? So just to segue into foundation models for biology. So how can we use generative AI for inverse design problems? So we have these large biology data sets. So for example, GDB has around 166 billion enumerated proteins, enumerated molecules. And we have, Sync, ChemMPL, PubChem, and so on, which have like, now Sync has more than 1 billion molecules, right? So we can build a generative model, much similar how you would build a generative model for language. And then we can use some of the techniques that have been applied in natural language processing to generate new molecules that satisfy specific properties. So for example, you might want to generate molecules that are that have low toxicity and high stability, or binding to a specific target, that are soluble and so on, right? And then we can use the traditional in silico methods like molecular dynamics or docking simulations to prioritize the designs that come from the generative model. So this is one of the pipelines that we use. So we have a large amount of training data from to train an autoencoder. So an autoencoder is basically a machine learning model that tries to reconstruct the sequence of a peptide, right? So we we pass the sequence to the model, and the objective of the model is to reconstruct the peptide. So can it reproduce the peptide? And once we build this autoencoder model, so we use a variational autoencoder, we can sample from this model which will, so when we sample from this model, we are kind of generating new peptides from this. And then we have, this model also let us have access to a latent space, which is a representation of the model. And then the latent space can be used to fit a classifier on the model. So we can make predictions on the output of the model based on the representations from this latent space. And then we can use this to sample from the model, so that you can use something like a rejection sampling in the latent space, so that the molecules that come out of the sample from the latent space satisfy some of the desirable properties that are important to you. So coming back to the specific problem of antimicrobial peptide design. So our unlabeled data is from UniProt, Sysprot, and Tremble, and we took 1.7 million unlabeled data sequences. So one thing to remember is that this research came like many years back. So the kind of models that we developed will not be considered large by today's standards, but these were pretty large at that point of time. And then we trained an autoencoder using this unlabeled sequence of data, right? And this the samples from this autoencoder were used for control generation to generate a set of candidate peptides, and then they were screened using both deep learning and simulations. So deep learning was used to test for things like toxicity, so that we avoid peptides which are toxic. And then we tested it in the wet lab, and the outcome of this was two novel sequences with high broad spectrum antimicrobial efficiency and low toxicity. So one key thing to remember is that this entire pipeline, from training the model to wet lab evaluation, took only like 48 days, and the most expensive step in terms of the time was always the wet lab evaluation, and we can do all the screening and the generation and the filtering and so on very quickly. So in the bottom, the picture label C is what we developed called a peptide walker. So what this allows you to do is it helps you visualize as you make changes in the design of the peptide, like as you switch out certain amino acids, what happens to the properties that you are interested in, right? So this is kind of an interactive way for us to evaluate a generated peptide, and helps us to see like how these properties are changing to a more favorable condition as we explore the latent space of the peptides. So again, so the wet lab evaluation has shown like promising results. We discovered, so these are the two peptides. We got like 10 to 50% success rate with generative AI compared to less than 1% success rate with existing methods. This was some press coverage in the press for the antimicrobial peptide project. So now let's come back to small molecule design. So the question that we asked was, can we use the same approach for small molecule design, right? So for this, we built a very large foundation model, which we call like a Malformer, though the earlier version of this, the Science Advances paper, used a much smaller and a different model. So this is our latest model from IBM. We call it Malformer XL. It is trained over 1 billion molecular text strings in SMILES format with relatively limited hardware resources, around 16 V100 GPUs. It is trained in such a way that it is very fast. We used many tricks in natural language processing, like linear time attention and positional embeddings. It is the state-of-the-art universal chemical language model. We tested it on 70 plus molecular prediction tasks. And it shows emergent behavior in the sense that even though it is just trained from the text representation of strings, it can actually have some insight into the geometry in terms of bond connectivity and structure and so on. So again, this is some more insight into the Malformer model. It is trained on large chemistry data sets using the SMILES representation. And once we have this model, you can use this for many different tasks. So for inverse design or property prediction, retrosynthesis prediction, virtual screening, and so on. So some of the properties that we evaluated on is on this benchmark called QM9, where our method was very competitive compared to graph-based methods and methods which took into account explicit geometry. So these are mean average error, so the lower numbers are better. So we also tested it on many molecular classification and regression benchmarks. For example, toxicity, blood-brain barrier prediction, side effects, and so on. And we get competitive results in almost all of the tasks. So now if we go back to our pipeline for antimicrobial discovery, we kind of applied the same methodology for small molecule design. So we started with a molecular VAE, which is trained on SMILES. And then, so remember, we are trying to predict whether a small molecule is going to bind to a specific protein target, right? But interestingly, in our project, we did not use the structure of the protein at all. We used the embeddings which are trained just on protein sequences. Again, this is where foundation models play a big role, because the protein sequences were also trained on the sequences on very, it's a very large model, which is again trained in an unsupervised way, so that the embeddings that are the representations created by that model can be used for another task, in this case, binding affinity prediction, right? So we looked at two targets, MPRO and Spike, and then we used the same rejection sampling based approach to predict, to generate molecules for both of these targets. And then we used a screening method to rank these designs. So the rankings were done on both using machine learning models, for example, for predicting toxicity, but we also used other traditional methods like docking, right, to rank the candidates. We also used a retrosynthesis predictor to make sure that the molecules that we generated are able to be, we can actually synthesize these molecules in the lab. These generated designs were tested using rapid fire MS assay and front assay by the Diamond Lab. And finally, we got 2 novel hits for both Spike as well as NPRO. So again, the key points here are that we did not use the structure of the protein at all. And the entire pipeline was very fast. It took only like 4 to 6 weeks for us to go from building the model to coming up with the final candidates that can be synthesized and tested. And you can see how the number of molecules that we filtered go as it goes from generation to screening to synthesize and testing. So we generated around 800,000 molecules per target initially. This, after final screening, this was reduced to a top ranked 100 targets, out of which 4 per target was tested for synthesis and evaluation, and we found total of 4 hits. So these are the 4 molecules that we found. Another interesting aspect of our design, which we think was kind of very instrumental in the success of this project, was retrosynthesis prediction, because very often what will happen is we design a molecule and then we send it for synthesis, and then they'll come back saying that there is no known path to synthesize this molecule, right? So what we did to solve this problem was we had our own retrosynthesis prediction model, IBM-RxN. Along with all the molecules, we also sent the predicted pathways to synthesize this molecule. And for 5 out of 8 molecules that we sent for synthesis, the predicted pathway by our model was chosen by Enamine, our chemical synthesis partner. And for 3 of them, they identify a slightly different pathway. So you can see here the predicted pathway as well as the actual pathway used by Enamine. So these are some results from the neutralization assays. So we also immediately after our generation and our screening process, we released around 3,000 molecules to the public, where they could look at each of these targets and then filter these molecules based on many different criteria. So for example, synthetic accessibility is one of the criteria, novelty, solubility, selectivity to this particular target, toxicity. And then you could basically use this web interface to select the molecules, download these molecules, and then you can do your own synthesis and analysis for each of these molecules. Another important thing to highlight here So this is a press release that that came out in April of 2020, right? So if you look at the timeline for SARS-CoV-2, the disease was first identified in the end of 2019. The first lockdown was, if I remember correctly, around March of 2020. By April 6th, we were able to release this portal and the first set of molecules that could potentially be useful in this search, right? So within a month. And this was possible because we had all the pipeline set up. And the only thing that needed to be changed in our pipeline was the actual protein target. And we don't even need the structure, right? We just need the sequence of the protein target, right? So that's why I think this kind of method is very, very useful, because it is quite fast, and it is very useful in a situation where we need to identify a set of candidates really quickly. And of course, the hard, challenging task is to validate these designs, right? So that always takes more time. But the initial stages of identifying a good target and removing, a lot of candidates because they're not suitable because of toxicity, solubility, synthetic accessibility, and so on, that can be done relatively quickly. So to summarize, for this project, we did not use protein structure at all. These were the early days before AlphaFold 2, right? So we couldn't use AlphaFold to generate even a candidate structure. So we used foundation models for many aspects of the discovery pipeline. So for example, for generation, for toxicity prediction, for retrosynthesis prediction, right? So foundation models in general are, I feel like, very useful for many of these design tasks. And of course, wet lab validation is very important. But the real advantage of using foundation models for this kind of task is that it's a very fast pipeline. The initial stages of the discovery process can be completed really quickly, and then the wet lab evaluation, the design molecules can be passed for wet lab evaluation really quickly. So that's all I had. So I'd be happy to take any questions and hear your comments.
Okay. Thank you so much, Ilaria, for your beautiful presentation. I see questions coming up. One is from Oliviero Carugo. He asks you, might the use of protein three-dimensional structure information improve the success of this pipeline?
Yeah, I think so. Definitely, it can improve because there is a lot of research now coming up that sequence-based models are not as effective compared to structure-based models. But again, when we started this project, the structure was, first of all, not readily available. So the advantage of this method is that it can be used even when the structure is not available. The other complexity with using the structure is that structure-based representations for proteins are a bit more complex because you need to have like a 3D model. And with a sequence-based model, we can leverage all the advances that we had in natural language processing and kind of use it directly in our model. So that's some of the advantages of using a sequence-based model, but definitely structure could improve the accuracy of the predictions. The only challenge is in the complexity of the modeling.
Okay, we have another one from Melissa Mantonico. This is about using experimental screening data as a trained set for predicting promising inhibitors. And also the problem of using a small training set is always a problem in the sense that it can lead to overfitting of the model. And in this case, how do you tackle this, and how would you integrate experimental with the AI-driven drug design?
Right. So this is, I think, a very, very good question, because at that time when we started this project, we did not have any affinity data for SARS-CoV-2 at all, right? Because this was early stages, and there was not any data at all on specifically for Mpro or Spike. But what we were able to design, what we were able to leverage was the affinity data from other targets. So what our model learns is not specifically whether a particular molecule can bind to Mpro or Spike, but it learns whether a particular protein can bind to another small molecule, right? So it is learning much more general. And for that, we have a lot more data. It's not huge, but we still have around, like more than 100,000 labels, right? If you take any protein target and a ligand, we have binding affinity data. So that's another advantage of using a foundation model, because now we don't need that much data about a particular target or for a particular molecule, right? Because we can generalize from the learnings that we have from other targets and other molecules. So we are kind of learning a generalized model, which is trying to predict whether a particular protein will bind to a ligand. So that way, it was not, much of a problem for us, though we could, of course, use, if we had the data that we have right now, we could have done, much better, because it would be much more specific to the target of interest.
Okay, we have time just for two quick questions that I think are important. One is this one by Pradeep Nagaraj. And this is about when you are dealing with undefined structural models, if you have models that are not accurate, or if you have questions, would this affect your predictability in your model?
Yeah, if I understand the question correctly, if you can't resolve the structure properly, or if the prediction model is such that it is not able to predict the structure correctly. So in this case, we are not trying to predict the structure, right? We are trying to learn an embedding from the protein sequence. So we are not directly predicting the structure. But I think to generalize your question a bit, like for example, if we see a protein that is very different from what we have seen before, right? So then how will the model perform? I think that's an interesting question, because in our case, I don't think we had a similar molecule to SARS-CoV-2 in our data set. But it still was able to make reasonable predictions. So I think that again goes back to generalizability. And I think it's where one area where the foundation models really shine, because what it allows the model to do is to generalize better, even if the actual data that you're testing it on is kind of very different from what you have seen before. So yeah, to answer your question, I think the accuracy will be lower, but we can alleviate some of those problems by building these large models in a self-supervised way.
Okay, and the last question, it's about explainability. Would you care to comment? This is from Daniel Kazenpour.
Yeah, so typically deep learning models were thought of as black box models, where it is very hard to understand what's going on inside the model. It makes reasonable predictions, but we don't quite understand what were the things that it locked in on to make those predictions. So we can't go back and say, this molecule binds better to this target because of these residues, and so on. But I think over the years, we have made a lot of progress in terms of explainability and to understand how these models work. But I think that's one area where we need a lot more research, I think.
Okay, thank you very much, Inara Vigil, for presenting and for answering these interesting questions. It was really a pleasure. I also thank all the speakers, Beatriz and Thiago. And unfortunately, all good things come to an end. But if anyone from the audience still wants to address some unanswered questions or wishes to contact our speakers, please feel free to do so. As we end, I would like just to call your attention for the next Instruct Structure Meets Function webinar, which will be hosted by Grenoble. And also, for those interested in structural biology, we will be hosting the biennial this year at the end of May from 22 to 24, and that will be in Cascais, Portugal, by the ocean. So please join us and continue the further progress in structural biology. Thank you so much, everyone. Hope to see you all soon. Bye-bye.